So now we'll discuss how you can use things like QQ plots to really detect problems like population structure or hidden ethnicity in your genetic data. And so in the same way that both the problems of winner's curse and the problems of hidden confounders or data heterogeneity arise because you're testing lots of SNPs and you typically need lots of individuals in your study. In the same way, the fact that we do so many tests makes available to us a number of tools that allow us to detect these problems and then correct for them. So for example, here's a QQ plot. So the QQ plot again is very important classically in GWAS studies. And so again, typically the first plot that people make when they do a genome-wide association study is they draw the QQ plot. And so again, if you're on average for the majority of phenotypes that you might look at, particularly complex organism level phenotypes like disease incidence, you generally speaking expect that of the millions of SNPs you test, only a small proportion of them are actually truly associated with any particular disease or complex trait. And so when you test 10 million SNPs and you expect most of the null hypotheses to be true, then what you expect is a QQ plot where the line is fairly close to the diagonal. And so those are represented by the green lines in kind of near the diagonal of this plot. And so one thing you should know about this QQ plot is that in comparison to the QQ plot I drew before, the X and the Y axis represent negative log 10 p-values, whereas I think before they represented p-values from 0 to 1. And so the idea is the same. Your QQ plot should look close to the diagonal. It's just that I want to point out that the X and Y axis are transformations of the p-value. So this, again, lines that are close to the diagonal suggest that there's not a lot of signal in your genome-wide association study, which is generally perceived to be a good thing overall. Whereas strong deviations from the diagonal, so that's represented by the black line, are usually indicators that there could be potentially confounders in your association study that you didn't consider before. And so when you perform an association test, it's very frequent that the line that you get would be one that looks like a black line. So in this case, typically what you would then do is you would then try to correct for both kind of known and unknown confounders, and do so in a way such that you basically keep trying to correct for different confounders until your black line essentially moves closer and closer to the diagonal until you deem it such that you are not essentially over predicting too many SNPs as being associated with your given trait. And so the next question you might have is, well, if things like population structure, ethnicity give you a lot of confounding for association studies, then why not just correct for it? Right, and so correcting for things like population structure and ethnicity is problematic because on previous slides I showed you that one particular approach to addressing known confounders is that, well, if you know what the gender of each individual is, then you can use these kind of nested models to correct for gender. And so in a similar way, you might ask, well, if ethnicity is a problem, then can't you just build a nested model to address differences in ethnicity? And so conceptually you could, in the sense that if you recorded the ethnicity of every individual in your study, then suppose you're doing a study of BMI and you wanted to correct for age, gender, and ethnicity, then presumably you could build two models to do this nested approach, where in model 1 you just fit a line for age, gender, and ethnicity. In the second model you fit a line with some weights for SNP, age, gender, and ethnicity, and you could compare the two and then test to see whether your SNPs provide more explanatory power on top of what age, gender, and ethnicity can tell you. So the problem is that oftentimes it's actually really hard to record ethnicity as kind of a variable that you can correct for in this kind of framework. And the reason for this is that, especially nowadays when you see a lot of admixture between different ethnicities. And so you have a lot of individuals that are, for example, of mixed population. You might have somebody that, is like 25% Asian, 8% Irish, and whatever, so on and so on. It's really hard then to actually, number one, get a good estimate of what those different, percentages are. And number two, it's just, it doesn't work when you're dealing with populations, when you're dealing with cohorts of like millions of people. Getting good numbers like this is hard. And finally, even if you could correct for ethnicity by getting really good estimates of how much mixture there is, within each individual, you still would have a really hard time correcting for things like the fact that people within the same family share a lot more genetic similarity on average than people between different families, and so on. And so we basically need a different way of getting a good estimate as to, what a person's ethnicity is, or, how genetically similar people are overall, as opposed to for a given SNP. And so here's where visualizing a so-called genetic similarity matrix kind of really helps at least kind of visualize what the structure, population structure, and family relatedness of your cohorts look like. Right. So a genetic similarity matrix can go by a number of different names, but essentially what a genetic similarity matrix is it's a heat map where the rows and the columns represent the different individuals in your study, and they're typically in the same order. And the heat map basically tells you what the essentially correlation in the SNPs between any pair of individuals is. And so, like most heat maps that we've looked at in this class, the diagonal is very prominent, right? And so white in this diagram means two individuals are perfectly correlated in genotype, and black means they're not correlated at all. And so, as you'd expect, the diagonal in a genetic similarity matrix is white because each individual is perfectly correlated with themselves. I mean, that kind of makes intuitive sense. But then basically the structure of these heat maps start to look more and more interesting depending on what the structure of your cohort is. And so if, for example, you were looking at a cohort in which you have three broadly related groups of individuals, so say you have like three ethnicities, say Europeans, Africans, and Latinos, then what you would generally see is kind of like three largish blocks on the diagonal. And those blocks correspond to the fact that individuals of the same ethnicity tend to look more similar to other individuals of the same ethnicity than to individuals of other ethnicities. And, we're all humans at the end of the day. And so obviously there's still a lot of correlation between people of different ethnicities, but generally speaking, depending on which SNPs you're looking at, individuals of the same ethnicity will tend to look more similar to each other than to people of other ethnicities. And so when you have three ethnicities in your cohort, you might get three kind of loosely defined blocks on the diagonal. On the other hand, if you have, for example, a lot of nuclear families in your cohort, but your cohort is of the same ethnicity, then the kind of heat map you might get would look like the one in the middle, where by and large the most prominent feature of this heat map is a diagonal. But if you kind of zoomed in on the actual heat map, what you'd see is small little more prominent squares on the diagonal, and each prominent square would typically correspond to a nuclear family. And so the squares on the diagonal of a family, of a kind of a family-driven genetic similarity matrix, would be more prominent than the blocks on the diagonal of an ethnicity-driven similarity matrix, because a parent and a child will tend to share much more similarity, relatively speaking, than two unrelated people of the same ethnicity. And so finally, in your best case scenario, where you have no family structure and no ethnic structure in your cohort, then the pairwise correlation matrix you would get is basically just a white diagonal with more or less blackish background. And so that would correspond to having no population structure in your cohort. And so the idea here is that genetic similarity matrix is, number one, a way of kind of visualizing quickly what the structure of your cohort is for your genetic association study. And secondly, it intuitively forms the basis of the inputs to basically different approaches like principal components analysis that can help you actually convert these kind of similarity matrices into covariates that you can then adjust for using some of the nested models that we talked about before. And so, although on the next few slides we'll be talking mainly about the principal component analysis approach, there is another approach which is more powerful called the linear mixed model, which can correct for features of these similarity matrices that you see in a kind of a stronger way than principal components analysis.